This is, what is this? This is 7-10, which has to do with mitosis. Mitosis is the process by which cells go through in order to duplicate their DNA when they need some extra. Why do they need extra DNA? Well, for a couple of different reasons. Why do they need to make extra cells? Again, for a couple of different reasons. So, let's take a look. When do cells replicate their DNA? During mitosis. Specifically, if you remember, we learned about something called DNA replication. DNA replication is when we take our 46 chromosomes and turn it into 92. And so that's when DNA helicase and DNA polymerase comes along and starts adding new nucleotides to the chains and then make, oh, my cat can't get in the kitty door. Come on, kitty. You can do it. There's a cord in the way and he thinks it's blocking the door. Come on. Yeah, it's not that bright. So, in order to do this, cells need to undergo the process of mitosis. Now, cells also need to make more copies of themselves for a couple different reasons. One is to grow. You all start out as one cell, and we end up with trillions of cells. Something on the order of times 10 to the 10th cells, a lot of cells. And so, in order to get from one to that many, well, we need to make exact copies or duplicates of the original cell. Second of all, we lose a lot of cells due to just wear and tear, or the life of the cell happens to be short, so we need to make some new ones. And the last one is that we need to stay small because we need to be able to get food and good stuff in and bad stuff out of the cell quick enough, or else the cell could starve to death or die due to poisoning. Also, the bigger the cell, the greater the demand there is on DNA. So if we have a nice small cell, then the DNA doesn't have to work so hard. And so DNA is happy when he doesn't have to do that. Okay, so DNA, let me get that all the way. There we go. The cells themselves need to stay small. This is kind of going along that same idea. Cells need to stay small in order to move stuff like nutrients, oxygen, and water into the cell, and waste like CO2 out of the cell. Now, in a little teeny tiny cell, it's very easy because the middle of the cell isn't far away from any outside surface. But the middle of this cell is very far away from an outside surface, so it's much harder to get nutrients in and waste products out in a big cell than it is a small cell. So what do? Is we look for a cell that has a high surface area to volume ratio. The higher the surface area to volume ratio, the more likely it is the cell will survive. So if we look here at the surface area of each of these different sized cubes, we have 6, 24, and 96. And then if we look at the volume, we have 1, 8, and 64. So if I compare the surface area to the volume, in the teeny tiny one, we have 6 to 1. The medium one is 3 to 1, and in the big one, 1.5 to 1. So this cell is too fat for your body to be able to survive. So that's why our cells stay nice and teeny tiny, because it just makes it easier on the cell to survive. Okay, so our cells need to divide. They need to go through mitosis. How do they do that? Well, they need to first double up their strands of DNA. So we have 46 of these little strands of DNA called chromatids. But when a cell is about my cat still, come on cat, you can do it. Yeah. Yeah, but I have the door closed so he can't get in anyway. So I've got these chromatids. I got 46 of them. But in order for a cell to divide and maintain that level, I need to double it up to 92. So that way when each cell splits, they each get their complementary 46. Don't even cat. So how do I do that? How do I turn it into 92? Well, that's the whole DNA replication that we learned last year right before Christmas. So when my cell makes an extra copy of DNA, it sticks to the original. So here's my original strand of DNA, and then here is my little photocopied strand of DNA. So I have two strands are identical to each other, they're duplicated, and each of these chromatids is now referred to as a sister chromatid because of the fact that they belong to one chromosome. So if I have a chromatid over here and a chromatid over here, these are not sister chromatids because they do not belong to the same chromosome. It's only when they belong to the same chromosome do we call them sister chromatids. And they're held together in this area right here, which is called the centromere. It's kind of like Velcro, and it holds the two together. The reason why I call it Velcro is because it's very easy to rip it in half and let one guy go that way and one guy go that way. So it's a kind of a weak area where the two chromosomes or the chromatids are held together. Now, in a dividing cell, there's going to be a couple things that we see. And these are the churros that we talked about back in Chapter 7. And what they do is they just kind of help. Here comes the cat. They're going to help pull the DNA apart. So here's my chromosomes, and they're kind of on edge, so they're little sideways X's right now. There's that centrosome that we were talking about, or sorry, centromere, right there, where they're being held together. And so what they're going to do is the centrioles are going to throw out a line, and there's the cat. I hate you, cat. So they're going to throw out a line, kind of like going fishing for chromatids, and then they're going to grab onto one. So those fishing lines are called spindle fibers. They're just microtubules and microfilaments which pull chromosomes to opposite sides of the cell and separates them in half. But the only way that they can grab on is due to this thing called a kinetochore. So if I look at a chromosome up close, there's a little hooks on either side. That's, stop rubbing the microphone, called the kinetochore, and that's where, stop it, the spindle fiber is going to reach out and grab onto that. So we kind of need to know about these things in order to understand how mitosis occurs. Alright, so let's take a look. Oh, that didn't fit. Hold on. Scoot. There. I don't know why. Oh. Funny, it's different than the one at school. Oh, bummer. Make that smaller. Sorry about that. Can't get off the microphone. Hey. So the cell's life comes in two main stages. We have the first stage right here called mitosis, and then the other three sections right here, which is called interphase. So here's stage 1, stage 2, like that. And each of the stages are separated into little smaller stages. So get off the chair. So the first one, interphase, which is this big blobby right here. Interphase is when a cell is chilling. So when we look at a cell under a microscope and it looks like that, that's a cell in interphase. But it's not just chilling, it's actually doing some important stuff. In the first part, in G1, the cell is actually growing, because when a cell splits in half, it makes two teeny tiny cells. And so they want to be grown up like this big guy right here, so they need some time to grow. So that's what's going on during G1. That's going to be the growth of the cell turning into a big old grown-up cell. Then, if he's going to divide, he needs to double up. So I've got 46 chromosomes here. He needs to double up. So there's 92. So during the S phase, that's when the DNA doubles up and is now committed to go ahead and divide later on. So then over here, in the third phase, G2, he still has 92 chromosomes, but now he's kind of mentally preparing himself for the split, for the move. Once he gets to the end of that, he is going to go into mitosis. Mitosis is when the actual nucleus and the DNA do their dividing, and it's split into 5 different phases. P, M, A, T, C. Don't pee on the mat, you see? So these are the phases of mitosis, and you can see them here: prophase, metaphase, anaphase, telophase, and then sometimes you'll see cytokinesis kind of shoved in there like a little wedge. So this is all the M phase right here, made of these 4 phases. Now this is just a shorthand of what happens here. I'm going to show you the longhand of what happens next. Okay, so mitosis begins when this thing called cyclin says it's time to begin. So the cyclins are something that can be produced by the pituitary gland in your brain that can tell everybody to get going. So cyclins say, okay, time to begin. First thing that's going to happen is prophase. Prophase, pro means first, and phase means, well, phase. So the first thing that's going to happen is that the nucleus, which once was a nice solid structure, is now going to start to, oh, that's really big. It's going to start to get little holes in it like this, and it's going to fall apart until eventually there's so many holes that the whole entire thing just disappears. So the nucleus disappears. Second thing that's going to happen is that the chromosomes appear. Remember, our chromosomes were in chromatin form, which is just a big old hairball. So they start to separate and start to coil up and turn into actual chromosomes. So chromosome, 4 chromosomes in there. Then the centrioles, these little guys right there, are going to migrate to opposite sides like that. So I've got one on one end and one on the other end. And the last thing is that the fishing line is going to go fishing for some chromatid. And so they're going to reach out and grab onto the chromatids, one from each side. Okay, once that happens, so these 2 phases right here, those 2 are prophase. Once that happens, we're going to go to metaphase. Meta means in the middle, so metaphase is all the chromosomes are going to line up in the middle, like that, and they're being pushed around by the little spindle fibers. So the spindle fibers are telling them to line up in the middle. After that, we're going to hit anaphase. Anaphase means away. And so as you can see, they're being ripped in half. So we had our little chromosome like this. So they're being ripped in half. So one chromatid goes this way, the other chromatid goes that way. And they're being dragged along the middle. So they're kind of being pulled in that direction. And so they folded in half because it's, that whole thing right there just folded in half. And so they're being pulled away from each other, away from that midline where they were hanging out not that long ago. Then, after anaphase, we're going to get to telophase, which is where the nucleus decides to show back up again. The little chromatids now decide to uncoil and turn into chromatin. And the spindle fibers, all these little guys, decide to disappear because they're done. There's nothing else to do. So I still have one cell, though, but it has two different nuclei. So that's how I know I'm not quite done yet. So we can see here's the new nuclei right there, and we can see that it's still one cell. So I need to divide the cell in half. I need to karate chop them in the middle. That's going to be done by the process of cytokinesis, which means to split the cell's cytoplasm. So what's going to happen is we're going to get this thing called the cleavage furrow, which is that little indentation right there. So it looks like a little indent all the way around. The organelles are going to be split. Some are going to go to one cell, some are going to go to the other. And then eventually the cell membrane grows inwards until it pinches off and you get two brand new cells. Then they just go back into G1, start all over again, grow and turn into a grown-up cell. Now cells are pretty smart. They know when to stop doing that when they bump into each other. So say you got a little cut like this and that little chunk of skin was gone, your cells would continue to grow until they bumped into each other, and then they would know that they're done. So that's kind of their signal. When they run into each other, that's their signal to stop growing. But every once in a while, some cells don't get that signal. And so we get a bad cell, bad guy. And he starts to grow and grow and grow and grow until, he gets his own blood supply. That's not good, because one of these cells can jump off and go for a ride and end up pretty much anywhere else in your body it wants to. And so that's what we call cancer. That's bad stuff. And so we got to kill those guys by giving them medicine that attacks rapidly dividing cells, which also attacks your hair and your stomach, which is why most people undergoing chemotherapy tend to be bald, because the chemicals are attacking their follicles as well as the bad guy there. Okay, so that's it for mitosis. So now you've got it down, and then tomorrow we will talk about meiosis, which is instead of going from 46 chromosomes to 2 cells of 46, what's going to end up happening is I'm going to take my 46 chromosomes and I'm going to make 2 cells that each have 23, specifically little eggies and sperms, so they can get back together and, take a guess, get back to 46. Alright, see you guys tomorrow. Bye.